Welcome back. I'm really delighted to be able to introduce the last of the main stage sessions beyond today of what's emerging from the pandemic, and to introduce, I think, one of the most remarkable projects that I know of in the creative R&D field. And what we're going to be talking about over the next hour and talking to the main protagonists of is a project called World in a Cell. And it's a beguiling title, and in some ways it's completely self-explanatory, although I'm sure it's going to get much more complex as we go along, because it is a project that attempts to build a complex, immersive visualizations of all the systems in a single cell in the human body, and not just any cell, but a beta pancreatic cell, which for those of you who know about diabetes, or may indeed know people who suffer from diabetes, it's one of the key cells in the kind of diabetic illness. But this is a remarkable combination of art and science coming together. So I'm delighted to say the first part of the session, I'm going to be joined by the two principal investigators in the cell, and then we're going to swap the team around, and some of the team at the World Building Institute from USC are going to walk us through and give us access to some of the world in the cell. So I'm delighted that my first two guests on this session are Alex McDowell, RDI, who is well known to some of you from Beyond. Indeed, I think Alex gave the very first keynote at the very first Beyond, which is only 2 years ago, to be honest. It's not that long ago. But Alex is obviously a hugely respected figure in the world of design and in film production and film design, feature films from Minority Report to Fight Club. But he's also a professor at the USC Film School, and that's where he set up the World Building Institute, which takes forward the design futures approach that a lot of us find so exciting as a way of bringing different communities together. He's also an advisor to our Audience to the Future program and bringing that knowledge to it. And he's joined by Professor Helen Berman. And Helen is a distinguished professor of chemistry and chemical biology. I think I've got that right, Helen, at Rutgers University in New Jersey. And she's also the founder of the National Protein Bank. So this is where, as X-ray crystallography was being used as a technique to elucidate how proteins, the shape and structure of proteins, this was, as it were, the archive, as I understand it, of official descriptions of what those proteins are. So Helen's a huge contributor to the field of cell biology. So the interesting thing first we have to ask about this project is, Alex and Helen, what is the origin that put the two of you in a room together?
Okay, I'm going to start. As you point out, my background is in X-ray crystallography, and I'm a structural biologist, which means that my initial training was in studying the shapes of large biological molecules. And unfortunately, every day we see such a large entity in the COVID virus, is an example. 50 years ago, almost 50 years ago, in 1971, we co-founded the Protein Data Bank, where the idea was to store all of the information about biological molecules. And at that time, I think we had 7. The reason we did it is that we knew that somewhere there was gold in those molecules. And many of you may have seen articles in the newspaper as of 2 days ago, where there looks like there's been a huge leap in being able to actually predict the structures of proteins based on what's in the PDB. So that's a really big deal. So I've always known, since I'm a student, that these molecules are really, really important and contain huge information. But I also knew, I've experienced how difficult it is to communicate that information to people outside of the field. And it's been really hugely frustrating when you try to explain all that to people. And I know that scientists as a culture, we're really not the best storytellers. That's not something that we do very well. in some cases, people deliberately do not want to tell a story for reasons that I never quite understood. So when I moved to California, I began to work on the Pancreatic Beta Cell project because I thought this is a way of bringing all this molecular and cellular information together and really understand a very, very important biological problem, which is how insulin is produced in the cell, and maybe in that way understand diabetes. But the other thing that I was intrigued by with this project was that the founder of the project, Ray Stevens, early on decided that it was really important to collaborate with the School of Cinematic Arts, specifically with Alex, to create a world in a cell and make an immersive experience, and in that way be able to communicate about the science. And so that project really, really caught my imagination, because I said, this is what I've been trying to do, and I've never found the right vehicle. And so we started working on the project. I joined a couple of years ago, and we needed to be able to figure out how to explain the complexity of a cell. And in the process, we realized that the scientists didn't actually fully appreciate the complexity of the cell. We thought we did, but then when we were trying to explain it to the artists that we were working with, we realized that we didn't really have the tools. And so in order to do that, we realized that we had to develop a whole new visual language. And that's where Alex came in, because who better than Alex to begin to explore how we would do that. And so at this point, I'll turn it over to Alex. They're, enjoyed a really wonderful collaboration. Alex.
Thank you. Yeah, as Helen says, the provocation here, which was one that was really outside our experience, was how do you take this esoteric language of molecular biology? And Ray Stevens was very clear that that has been preventing collaboration, not within the field of molecular biology, but outside, even with other biologists, certainly in the other scientists. And that this point that we've reached, as Helen says, this incredible complexity that's sort of within our grasp now with the technologies we have, it is not being explained, and as Helen says, by the science side, who are really not adept at telling stories. So the fantastic challenge and the provocation was, how do you take this esoteric language and how do you convert it into a kind of universal language, a language that would allow high school students and scientists and undergrads all to be able to gather around the particular issues and capabilities and door opening that getting inside the cell represents. So we started off with this fantastic scenario where we had, metaphorically, one side of the table of artists, engineers, architects, game designers, animators, the kind of visual arts media space that I'm familiar with, sitting across from a row of scientists, looking blankly at each other for about 6 months, going, we don't know how to speak to each other. I mean, it's really, we're in so much in our specialized fields, which is an issue for, as Andrew and I've spoken about a lot. I mean, it's an issue across all disciplines that this area of specialization doesn't allow us to communicate with one another. But the task here was to find the Rosetta Stone, basically, and say, we now have a common language. And that actually boiled down to origami. Both sides of the table completely gathered around the idea of we know about folding, structurally in the cell space, structurally in the architectural space. once we'd established that, this project really took off. And this has been, you know, since Helen joined in the last 2 years, we've moved from understanding the kind of structure of the components, the constituents of very specific molecules and proteins in the cell, starting to be able to model those, taking the pancreatic, the protein database data that Helen's been spending so much time developing, and turning it into architectural language. So we started off in a very manual way. We started breaking down the idea of what origami is, what its fundamental elements are, and we came down to a simple piece of geometry, a tetrahedron, which again was universally understood. And I think where we are right now is, first of all, we found the ability to create recognizable objects, each protein having a distinct silhouette, which really wasn't true up till now. And then assembling the component parts, the kind of Lego pieces, into a sort of infinitely malleable different types of recognizable objects that could animate, could exist in three-dimensional space. They're all dimensional, in fact, four-dimensional space, because we really, for the first time, began to see behavioral characteristics. So from my point of view, and from the point of view of the artist side, which I'll quickly break down, we were seeing something that we could understand as character, or something, components in a story, without in any way anthropomorphizing the complex science that we were addressing. We could see that it was possible to use storytelling language to see the relationships between these moving parts, each one that had an identity. And that as we began to establish that, we began to see that we could tell a story that was coherent and reflected the way that message was passed through from one side of the cell to the other, essentially, where insulin would come out. And so we've modeled about 30 proteins and constituents now. We've been able to demonstrate the way they interact with one another. And I think really importantly, we've been able to use technology that we're all aware of now. You know, we're intimately aware of virtual reality and augmented reality, but for the first time, placing these three-dimensional objects into four-dimensional space and associate geolocation and scale, you know, relative behavior through time and space in a way that you could enter that world. And this is where we get to the world in a cell. We have this complete world now that we are inside in VR. And for the first time ever, we can actually see the massive scale differences, for example, in the different constituents. So all of that has evolved over these 2 years, but the exciting sort of side research and side part of the project, if you like, which has become, actually surrounds the project, is this notion of art science. That in order to kind of reach across the table and get that Rosetta Stone placed in the center, we found that this magnetic center required a sort of integration, an entanglement of these two understood to be disparate disciplines, until we've created a third discipline, one that's called Artscience, as a single word. And really, this project's only been possible by doing that kind of intrinsically and establishing it deeply within the process. So I think, I think in a unique way, we've sort of gone a leap beyond what's understood to be art plus science into this single discipline. And I would say everybody involved in this project is now fluidly moving in a space that is singular, biological, architectural, and is, you know, proving out the idea that you can develop and collaborate and move forward really complex ideas gathering true science and true media and putting those things together. So
I'd like to come back to the art science thing before we introduce the team, but also there's so much in what you've both just said to kind of unpack, I think, to set people's expectations before they see the world in the cell. So one of the things is, I guess, you know, from sitting outside this, there have been, I'm interested in, Helen, how that's worked from the scientific point of view, and whether where you've got to is the expectations you had, because from the way you both describe it, is world in the cell, it's obviously a visualization in one sense, you know, there's a visual component to it, but it also seems to be a semantic project as well. So you've created a language, and so I'm wondering whether, you know, there's a lot of talk in the UK at the moment around digital twins and as a new way of thinking about simulations. So is it a simulation? Is it a visualization? Or is it a linguistic project, and this is the canvas for these?
It's all of the above, I think. Everything, it's all of that.
Okay.
And actually, one of the things that happened is that the artists would say to the scientists, you know, if they didn't understand something, they just assumed that the scientists would understand some concept, okay? And we would say, no, we don't understand it. We don't really understand what's going on at all. And then we'd have to do a dive in to see what it is that we didn't understand. So by doing this kind of project, we upped our own level of understanding of what we thought and everybody else thought we understood, okay? So that was a really revealing thing. And by being able to put all of this together, we actually found there were two different specific scientific issues that had come up that we thought were one way, but by putting it into this medium, we suddenly realized we didn't understand. There were a couple, and that's going to happen over and over again as you put everything together. So what tends to happen in science, as every other field, is people work on one thing at a time, and they understand it very deeply. And then you put the two things together, and suddenly you don't understand it again. Here we have everything together, and we have to keep understanding. So this, by having this new language and this new way of looking at things, we actually, the scientists, learned things that we thought we knew, but we apparently didn't know, okay? And by being forced to explain to our artist colleagues what we were doing or what we were thinking, that actually gave us new knowledge. And then what actually happened in one presentation we did a couple of weeks ago, one of the people who was involved in the VR started explaining things using language that he got from us and became part of him. And so he was using the science language completely comfortably, and without sort of having been taught in the kind of didactic way. It was by doing. So I think that this is a platform for collaboration, a platform for discovery, and it's also a platform for enjoyment. I mean, you look at something and it's fun to look at. So it's really opened up a lot of new doors for us as the scientists.
Yeah, we use a metaphor that I think remains appropriate, which is the idea of walking through a forest. So as Helen says, you're inside an experience. If a family walking through a forest perceive that forest in very different ways. If the mother is a scientist who is looking at mushrooms and fungi in the forest, then her focus is on that. If the father is just deeply interested in the evolution of forests and the scale of trees and the diversity, then that's his focus. If the older girl is doing a high school project, then she's looking at the forest in that way. And the young child is chasing butterflies. You know, all of those things are equally possible when you create this environment. And I think what Helen was saying about the challenge, it definitely goes both ways. I mean, world building, which is fundamentally the way that we apply this storytelling, you know, in 4 dimensions, and this is built out of our world building media lab, has been severely stressed by this project in the best possible way. I mean, world building is a young discipline, it's 20 years old, and it has traditionally, if you like, so far been mostly engaged in media language and storytelling. In this case, we had to rethink entirely what it meant to tell stories. And so the stressing of each other, I think, is like catapulted our disciplines forward as well. I mean, our ability to speak to one another is a piece of that. Our ability to take this knowledge away and this sort of rigor and start applying it to other fields is really interesting. And I think that where we are right now is that we've developed a kind of system of systems and are beginning to talk about urban planning in relation to this, and architecture and data visualization, for example. It becomes more and more relevant because as it stresses the system, we start applying it to other questions that world building is being asked. And of course, all of what we know now is being folded into that as really a huge extension of our capabilities.
And I suppose that within each of those, you know, and using world building as this way of disciplines touching, one might have thought that something like, you know, molecular biology would be one of the hardest places, but you found your Rosetta Stone in origami. Again, I'm not sure whether that's a metaphor or whether it's, you know, it's a visualization or whether it's a language. But do you think that to do this with other disciplines, you have to find a similar kind of Rosetta Stone, a point of origin to build out this language from, or do you think you've learned something more general?
No, I think we definitely approach every single sort of provocation that comes from, you know, very different fields, whether it's pure fiction, fantasy in the film space, as my background, or in game, or in future of engineering. We do have to find a sort of Rosetta Stone of this idea that if what we're really doing is to do with translation and language creation, there is a difference between creating a language in one space within one provocation and another, but the commonality is the way you think about it. So even if your Rosetta Stone is different, because of course folding is not necessarily the core Rosetta Stone in a game that's set 2,000 years in the future, and you use a different kind of logic base. This is about the rigor of systems design, I would say, and that underlying idea of how you develop systems design interlocked with storytelling, that's kind of the leap, I think. And in a project like this, you are having to question what storytelling means. You have to be able to say now, these characters have no agency, but they have an interconnected narrative. We rethink, and I actually believe that we found a new way of storyboarding for multimedia. You know, I think you can't tell stories in VR in the traditional way of that linear narrative has been told, and we've been forced to look at a different way of breaking down stories, and so I think we can take this knowledge and we can and are putting it back into a broad field, and it's really raised the stakes, I think.
And Alex, I know earlier you used the term art science for this, and obviously, you know, I think that you mean something different from sci-art, which was a thing that probably many people in the audience will have been involved in as well, which was art inspired by science or whatever. I'm wondering, Helen, this sci-art, this idea that there are common sets, you can create a common set of skills, maybe, particularly amongst some of the younger collaborators on the project. Do you feel comfortable with that? Do you think that's an exciting development, and that you can genuinely expect researchers in the future to combine knowledge across these disciplines?
Absolutely. I mean, I'll just say that in crystallography, which was my original discipline, there was always this understanding that there was some kind of communication between art and science, because crystallography is extremely visual, okay? And we've had to cope with how we do representation and how we look at things from the beginning. So that was sort of a natural evolution for me. But yes, I think that this is going to continue. I mean, it's something that certainly people in my field will appreciate very much, but I think it will begin to extend into other areas of science, as it is with the world in a cell.
That's really interesting. It suddenly made me think, of course, art and crystallography have a great background. The Festival Pattern Group from the Festival of Britain produced a whole series of designs which were inspired by early X-ray crystallography. So there is actually a long tradition there. And
yes, there is.
Some of the most famous photographs in history are actually X-ray diffraction photographs, yeah. I suppose, in an odd kind of way.
That's right.
So I think, does that bring us to a point where we can show the audience some of World in a Cell? Does that say that seems about we've almost hit our mark there, I think.
Yeah, so I think it's
sadly due to the constraints of the system. Alex is going to dip out, and we're going to bring in 3 of the collaborators from the World Building Institute. Now, Helen's going to stay with us, and we're going to do Q&A afterwards. But Jitin Singler, Brian Cantrell, and Alexandra Thermopoulos are going to now appear magically, I hope, by the power of our platform. And then I think Alex is going to start us off, showing us so that we can get some sense of both the complexity and the scale of the World in the Cell project.
Yeah, I think all that we're speaking about is relatively abstract until you actually enter this world and you realize that it's as Alan says, it's actually a highly enjoyable space to exist in and to fly in, to have fun in, and it's giving you back information as you go. But so I think what we want to show now is definitely beta. We're still in research, we're still in prototyping, but something that we'll have to ask the audience to imagine how it looks in VR, but that begins to express the scale and the beauty of what it feels like to be inside the cell.
I am hoping that we can bring up our 3 World Building Media Lab contributors, and then once Alexandra, Jitin and Brian are with us, then we can play out the first part of the World in a Cell walkthrough. Okay, I'm told that we're going to run a short sting whilst we affect this magical scene change and then come back to you. Okay, we'll be back in a second. Hi, welcome back. I'm sorry about that. That was entirely my fault, I think, in mucking up what would have been an otherwise seamless transition from Helen Berman and Alex McDowell. But I'm delighted now. We're going to see inside. Your expectations should have been truly raised and appetites whetted from that introduction. But now we're actually going to have three of the team at the World Building Media Lab who have created this project, who are absolutely essential to this project, walk us through. So I think Jitin Singla is going to lead us off, and then Alexandra Tomopoulos is going to then walk us further through. And I think Brian Cantrell is going to introduce us to some of the sonification aspects of Word in a Cell as well, and many other things. So Jitin, over to you. Introduce us to Word in a Cell.
Hey, thank you. So I'm Jitin. I'm a computational biologist and currently a postdoc at University of Southern California in Los Angeles. So World in a Cell, most of the aspects have been introduced by Helen and Alex in the previous panel, but it started as a collaboration between Pancreatic Beta Cell Consortium that wants to understand the beta cell technically in its entirety, and World Building Media Lab at Cinema School at USC. Right. So the main challenge that we had in hand was to understand the cell, and to understand the cell in a spatial and a temporal way, and also have some kind of new insights into how things come together when you put information from different scientific fields, right? So, and the way we chose when we started this collaboration with World Building Media Lab, the goal was to create a fully experiential virtual world in a single cell. And I personally believe that building such an experiential world is important for actual functional and insightful understanding of how life happens in a single cell. And I personally also understand it through an example, right? So if you want to understand how is it like living in a New York City, right? So you can have any amount of data about the restaurants, streets, landmarks, images, Google Maps, but to actually have a consistent and thought-provoking experience of how a life is inside a New York City, you have to live in New York City. And that's because you gather all this information, but you build stories, you build a narrative, and that's what guides your insights. And we want to take the same approach when we understand the cell. So we want to collect all this information that the Pancreatic Beta Cell Consortium is producing and that has been available in the literature, and use storytelling and world building to present it, right? So I'll give you one example of how beautiful when you put together the scientific detail and the storytelling and the world building perspective to it. Just one example of how things can change. So for example, if you consider a pathway inside a cell, so pathway is basically just a sequence of events that happens between different constituents of the cell, right? So you go from A to B, B to C, and so on. And for beta cell, one of the important pathways is how glucose comes in and insulin is released. And scientifically, it has, like the scientific community has been using this, if you see on the left in the background, this kind of directed networks to show just pure information, like arrows between different constituents of the cell. So what we did was we took this information and we presented in a subway map format. So a subway map connects with the larger audience. It has all the information that you need in terms of telling what component interacts with another component. And it has, apart from that, it also just have another geolocation information of major organelles. And inherently built in it, it also has a narrative, right? So it's what we call it's a 5D space now. And another example, so even if you look at a single pathway in this subway map, this metro lines, it feels like this is a linear pathway. But when you go deep into the weeds, you will realize that there's not just one copy of glucose or one copy of pyruvate. There are like millions of copies of each of these molecules inside the cell. So actually, it's kind of a non-linear pathway. And I think nobody has ever tried to show that visually or graphically. And we came up with something like this, what we call a non-linear pathway. And I think that happened because of this this art science collaboration. So, and I think next Alexandra will show you how we are also building new design language to construct different constituents inside the cell, which makes a crucial part of the world in a cell.
Good morning, everyone. Going to take off from Jitin further deep into the world of the cell. So one of the main goals of the project, as Helen and Alex kind of alluded to, was to create legibility in the world and give a kind of deeper sense of understanding, not only to scientists, but also a new, fresh look at science to a non-scientific audience. So because of the complexity of the cell, the rules of the construction of the world had to be very simple, but very rigorous. And the first rule that was developed was that the world had to be constructed using just one building block that, however, can scale infinitely and be assembled in a variety of ways. And the building block as a concept is a pretty universally intuitive one. Even from a young age, children are exposed to some variation of an object like a Lego block or other. And as we grow up, we recognize modular languages in a variety of capacities. In architecture, for example, in which I'm trained, we have brick building blocks. Or in molecular biology even, we have amino acid building blocks that make up proteins. And so as the design research lead and modeling lead on the project, I worked very closely with the scientists to understand the existing research and science in order to develop the modular language based on scientific knowledge and data. And after much iteration, the building block we ended up adopting was the tetrahedron. And this was a real milestone for the project, not only because it meant we could develop the language further and express it to the rest of the world, but because it was an object that all the members of the team could recognize from their respective disciplines, from architecture and engineering to molecular biology, where the tetrahedron is one of the most common forms taken by carbon, actually. So the tetrahedron as a building block became an accepted metaphor by all, and it really was a milestone in our kind of art science collaboration to be able to really get behind one particular kind of important aspect of the project. So the second rule was that the size, shape, and symmetry of each constituent must match that of the actual biological assembly of that constituent that it's representing. So this makes the recognition of form relatively easy for the scientists who study the constituents, but at the same time, the rule gives uniquely recognizable silhouettes that allow even those encountering them for the first time to understand and distinguish different shapes and start to recognize them as figures on the pathway that Jitin talked about. So on this slide, you see an example of GLP-1R, which was actually the first protein that we started developing. It's a fairly, it's not a very large protein. It's kind of normal size, and it has a kind of interesting complexity to it. So it was a good starting point for us. And you can see I've added on this slide different kinds of visualizations that came before us. So we have a stick model, a surface model, ribbon model, and our tetrahedral model. And each kind of visualization has a particular intent. The ribbon model was developed for structural biology to understand the structure of the helices. The surface model was more to understand size, shape, and symmetry maybe, and maybe points of interaction, because it gives you just the surfaces of the proteins. None of these visual languages were really adept. They weren't really made for virtual reality. So when you're encountered with the entire world of the space, if you were to see only surface models, you would get completely disoriented. It's not really a kind of protein protein visualization that you would recognize and be able to say, oh, that's that model. So a scientist going in will more easily recognize a tetrahedral model because it is kind of designed in a way to give you the unique signature that you can then kind of go through the rest of the world and kind of explore without having to continuously check what you're looking at. So these constituent recognition posters, inspired by World War II aircraft recognition posters, show those unique signatures that I mentioned of the constituents, and they're intended to kind of imprint on the users the silhouettes of the proteins that they'll be encountering in the immersive experience. And now I'll let Brian take over.
Hi, I'm Brian Cantrell. I've been on this project for a very long time, and I'm mostly responsible these days for the sound that you hear in both the VR prototype that we have out there in the world right now and in the upcoming generative version, which uses the actual PDB data to drive sound, both the sound and the structures. So because I do sound, it's kind of difficult sometimes. I don't have much to show you because it's hard to show sound, but I thought I'd talk a little bit about some of the issues that we've already spoken about earlier that Helen and Alex were talking about, and then also some of the things that we encountered in this collaboration that I think sort of get at this question of art science, Andrew, that you were talking about earlier or asking about. And I was very happy to hear you mention sci-art and compare the two because they are very different. But part of my own research and doing my own dissertation kind of attempts to tease out those differences. And so I'll talk about that a little bit, and then I'll talk about the procedural sound, and then we'll take a look at the video. So this diagram in the slides is just a basic tool that we use in world building to map out the world. And what it is just a way of looking at the world sort of from 30,000 feet and mapping out the systems of the world, any world that we're creating, and understanding how those systems interact to produce the world. Our theme is emergence, and this is how emergence happens in world building. So you have these basic rules, these basic systems that interact, and the world emerges from that. So in some ways, what we do isn't so different from science, which attempts, as Helen said earlier, to kind of tackle one problem at a time very deeply to create an image of the world, a representation of the world. Scientists do that with law-like statements, and they try to uncover physical laws. We as world builders, we live mostly in the manifest world, which you see on the right side of this slide. And in the manifest world, we don't have to ask, what is the theory of a staircase, right? Everybody sort of intuits how to use a staircase from the time you're a child. Same with opening doors, the same with empathy. And design, this is the world that designers inhabit, especially sound designers. Sound designers are dealing with a material, with a medium, in relation to which most of us have sort of a kind of peripheral, it's a very peripheral medium. So there's a lot of information coming at you every moment in the sound environment that you don't really think of as information. You're just immersed in it all the time, for those of us who have average hearing. So science has its particular aims and outcomes, and design has its aims and outcomes based on these two images of the world, the scientific image, manifest image. And I think what's exciting right now in the space that we're working in art science, and particularly with this project, is the idea that data is becoming a kind of medium in itself that glues these two worlds together. So in my own work, doing the sonification for the project, you know, the challenge really is, how do you take these stockpiles of data that we have piling up? I mean, it was interesting to say, you know, to hear Helen say earlier that when she started, they had, what, 7 molecules or something. Well, now there are like more than any researcher can ever possibly hope to keep up with. The data is just stacking up day by day, and this is in every field, not just in structural biology. And so for myself, as someone who designs immersive experiences, the question is, how do you use the manifest space, the manifest world, and especially the immersive space that, you know, we're all kind of inhabiting every day, but then you go into VR and you're in a container, a kind of immersive container. How do you use that to express data in a way that is ambient and not just about trying to accomplish specific kinds of tasks? And this is especially important for sound. So, you know, sound in a VR space is sort of what glues the immersive quality together, but it also is something you don't want to be in the way. You want it to give you lots of information, but not to get in the way of the visual experience. And so there is a kind of peripheral quality to sound often in these spaces that makes it perfect for expressing certain kinds of data sonically. But it raises all sorts of interesting questions about, you know, how that might apply to solving particular kinds of problems, or, you know, is data visualization or data sonification only relevant in cases where you're trying to perform a specific task or ask a specific question? I think those are big open questions, and there are lots of labs that are investigating that. And I think that this project in particular is moving into a phase now where we're better positioned to explore that kind of possibility for expressing data as an immersive sort of medium in the environment. So with that, I think we can move on to the demo, and the demo, I believe, is from the VR experience that we've already completed. So there is sound design in this demo, not so much sonification. That's all in the newer phases. So we can transition over to that.
Hi, so hopefully we're all here now, Manifest, and you've had the walkthrough from Alex and from Alexandra and from Brian and Jitin, and also seen finally at the end there that extraordinary visualization that brings together in the VR a sense of what World in the Cells achieved. So I can see we've got quite, as you will be entirely unsurprised to know, lots of questions here to go through. From the audience that have been prompted to me, and thanks to the team for that, for pushing those through. So I think let's start with one of these questions here, and I don't know who wants to take these, but maybe this one's, no, I won't say, I'll let you all dive in. Did you encounter any difficulties between the scientists and the visual artists when collaborating? I mean, we had at the start, I think Helen and Alex described it as a kind of 6-month standoff of mutual incomprehension. What, you know, how did they finally work together? But how did you break that down? And what were the difficulties? Did anybody just not like the project from either side?
We're not going to give away our secrets, are we?
There was a degree of laughter there, Helen, I think, from everybody. So
well, I think it was a process, and we had to get to know one another and learn how to talk to one another, and eventually we had a team that could do that. So the team sort of evolved and changed, and there were some people who didn't actually know how to do collaboration with anybody. And then there were other people who were hyper collaborative, and we just kept trying because we wanted, there was a core of us, some of the core are here, who really believed in this and wanted it to work. And eventually it did work. I don't know what, you know, maybe Alexandra wants to say something about that.
I think you kind of touched on an important point. We really had to get to know each other and trust trust the processes of each other and understand where each of us were coming from, and know that, you know, the knowledge of Helen was safe in my hands. Helen and I actually have been working very closely from the beginning in developing this language. So we went through all of the iterations together. And yeah, I mean, working with her has been amazing, but it is still a process to understand, for Helen to make sure that, you know, I understood the science, and for her to trust that I was going to be careful with it and represent it accurately in the world.
Jitin?
I'll just add to that. Sorry, I'll just add to that I was in the initial meeting for this project like 3.5 years ago, when there were, Jitin was there too. There were about 30 people crammed into a room talking about the potential for this. And I think at the end of that meeting, everyone was convinced it could never happen. but over the next few months, we realized, okay, there is a way to do this, and it required some adjustment. I think, you know, each side of the table had particular preconceptions about what it is to do science or what it is to do design. So there was a lot of kind of, you know, meta issues floating around, a lot of assumptions we were making about each other. And it took a while to get, you know, some of the designers to understand that science isn't just banging rocks together, and vice versa, you know, that design isn't necessarily unscientific, that it has a particular rigor to it. And it just took a while to get there.
That's a very good point, that one of the things that was eye-opening to me as working on this was all the rigor involved in the artistic process. I just didn't know that. And that changed my view a lot about what really goes on. Jitin, did you have a thought?
Yeah, I mean, just like referring to what Brian said, I entered 3 years ago into this meeting as a second-year PhD student, and I was like, why I'm going to this meeting? What's going to come out of it for me? I should focus on my research. I entered that meeting, and I guess it's been 3 years and I never left. And I don't think I can ever leave, because apart from everything that is going inside the project, like World in a Cell, to me personally, I think it was a huge facilitator for my graduate training. It helped me a lot to provide different lens to my own research. And I think one concrete outcome that at least has come out for me is like, I am able to present my research much better. And I think that happened because I had to interact with people who were coming from no cellular biology or molecular biology background. So it provoked me to present in a way that everybody can understand. So I guess initially there was some obstacle, but I think over iterations and talking to each other, and me, Helen, Alexandra has done so many just sit-throughs. Anytime we'll be like, okay, are you available now? Are you available now? And I think it took, apart from the meetings with everybody, it took a lot of those one-to-one interactions that brought us together on the same platform.
Fantastic. We've got another question, slight change of tack. It's quite specific. So this is from Professor Marian Ursu, who leads the Digital Creativity Lab at York University. So Marian is asking, could you elaborate on how storytelling, so he's emphasizing storytelling, can be employed as a tool for scientific research and discovery? He says it's clear how visualization and simulation, for example, could be such tools. But storytelling, question mark, slight sense of skepticism there from Marian, although I think he's not really that skeptical.
So I hesitate to say some of this, but I think I'll try. There is a point of view among some scientists that if they make their presentations very obscure and difficult to understand, it will make their people think they're very smart. Okay? That is a perception among some scientists, not the best scientists. The best scientists, Richard Feynman being an example of a stellar physicist who made a point of being able to explain complex ideas in straightforward and simple ways. And he was a great storyteller, okay? But in general, I would say most scientists get into their heads that they shouldn't make themselves as clear as they should. And then they don't understand how we get into the mess we have gotten into with the pandemic, where people are not listening to science. So I think that there is a culture, and I'm not sure where it came from, and not everybody subscribes to it. I certainly have never subscribed to it, but there is a culture that says that that if you're very obscure, that somehow, and you stay in your own little world, that's somehow better. But it's not, okay? It's just not. So, at least in my view, it's not. But there are enough of us who believe that we have to be able to explain what we're doing. And some people think that if you can't explain what you're doing and tell the story in a coherent way, then maybe you don't actually understand what you're doing. You, scientists, don't understand it. So there can't be anything that's that complex that cannot be unfolded as a story. So I think that there is a cultural thing that I have never quite understood.
I'll add to that. That's a great question. First of all, it's a great question, because I think this project has, for us who make storytelling, that's our living. This project really forced us to kind of, in a way, redefine what storytelling is, or what it can be, because you're dealing with the space of the cell that doesn't have agency in the way that we typically like. Agency isn't a thing at that scale. So you can't have protagonists really without anthropomorphizing, which we didn't want to do. So there were a number of challenges, and I think one of the ways that we sort of rethought storytelling was to simplify it. And I always think about the Russian constructivists, the experiments they did with telling stories with squares and triangles, and that was a kind of anthropomorphizing they were doing. But I think that what we were doing was something similar. And thinking about storytelling in a really reduced way. So in the way that maybe a product designer might think of storytelling. And yeah, I think that it really just has to do, it goes back to sort of what I was saying in my presentation before about, expressing things in a way that can be intuited by an embodied person in the world. Because what you're trying to express at the scale of the cell is counterintuitive in the extreme. And so the storytelling becomes a way to translate those highly counterintuitive processes and structures into a language that we can just sort of, our embodied perspectives can kind of understand and draw it together into a narrative.
I think that's really helpful. And actually, you've answered, I think, part of another question that Ivan Isakov was asking about what the relationship between observation and participation within the cell. So that's great. Marion also would like to point out that he was being hopeful, apparently, not skeptical. I've misinterpreted it.
I'm skeptical constantly. So
I would like to just very quickly give an example. As Helen mentioned, and in my slide also it came out, like a pathway inside a cell, right? So if say a pathway is 30 steps, whether a scientist wants to believe it or not, a scientist can absorb that information as 30 different interactions, but at the end, it's a story that starts from A and goes to Z, right? And if you look at the whole cell, there might be millions such interactions. So what you're going to do, you're going to remember million interactions you had. At the end, if you want to have a consistent picture of the cell, it would be a story that tells all these interactions. So for me, it's storytelling is basically an integration of data, right?
Okay, that's a new. I was just going to write that down actually, so that I could use it later. Storytelling is an integration of data. Fantastic. Now, I know that you've also, as well as using and working on the project, you've also taken this project out to educational audiences, both undergraduate and I think maybe at school level as well. So we've got several questions that are asking, how do you find that? How do you find audience are engaged by the abstract visualization? And also, what do you think this kind of project can do to bridge that art, science and arts gulf that happens certainly in the UK in education and probably, I guess, in the US too?
So I can start by saying that I use this as part of a high school course in Pasadena in Los Angeles to try to teach the students about molecules, but use this as the platform to do that. And they were really engaged, and they really learned a huge amount by working with this. And so I think this was a particularly talented group of students, but I think having tried this, I think we can refine methods to go into different kinds of schools. I think, Alexandra, you did things with very young people at the library last year. Maybe you could explain what you did.
Yeah, we attended, it was called the Meet the Makers, that's what we're in now. It was a mini maker fair at the Los Angeles Public Library. And the audience there was primarily younger people, a lot of, under the teen level. So we really had to think about what were some of the things, what were the takeaways that this younger audience could have, because some of the biological concepts maybe were a little too complex for them. And so one of the things that we did with them was an activity of paper folding, and we made little paper folded tetrahedra, and they seemed to respond really well to that, just the idea of having a building block and then thinking about the ways that you can piece them together to get a variety of shapes. We also had a desktop build of the virtual reality experience, and it was very visually stimulating for young and older audiences. There were a lot of parents accompanying the children, and they really enjoyed it as well. And we had some people that came through that were diabetics themselves and talked to us a little bit about their experience visualizing the information that they had been kind of taught from their physicians in a different way.
Yeah, I think it's really interesting that that this can engage at all of those levels, even though it's actually been produced not as an educational tool, but as a true kind of collaboration, working at the top. One of the things I think running through is people asking, so where can they experience this? So maybe that's a question. There's a question now, but where could we experience this? So it's come up in another session that I was running earlier on, that actually a very complex set of LiDAR scans, what everybody would like to see is a room scale experience where they could be surrounded by this socially. Have you got plans for World in a Cell that will take it into other spaces?
Yes, we would like to do that. We have to have enough resources to be able to do it, but that's what we want to do. Yes, definitely.
Yeah, we have prototypes in VR. You saw some of the video from that, and I'm not sure where. I think that is still kind of in the beta well, probably even alpha phase at this moment. But I think, yes, I think eventually the plan is to push that out to a platform like Steam or something like that, that people can access it. One of the issues as we move into the data-driven phase of this, where the models and the sound are being driven by data, is that we have to integrate pipelines for allowing the data to be put into the system. And so that's unlike a number of kind of ordinary VR experiences that you would just download from a platform. So it has a particular set of requirements that we're trying to work out. But yeah, it will be in the world at some point, relatively soon, I would think.
So I've got two last questions. One is, the first one is, a lot of the people in this session in the audience will have woken up this morning or yesterday morning, I think it was actually, to the DeepMind's announcement of their AlphaFold. So I guess they'll be asking, like me, is where does that sit relative to World in the Cell and all the work that Helen's done over all these years around protein structures? It's just an opportunity to see if you can bring them together, because I don't have the knowledge to.
So I can go and, so AlphaGo is basically predicting the protein structure, right? And it's doing that by learning from all the existing, I think 170,000 structures we have in the PDB. And
right, so that's something people may not understand, is that
so in the
database is actually the training set for the AI.
Yes,
yeah.
Okay,
yes.
right. No one explained that this morning.
I know, but
so there are two kinds of predictions that goes in protein folding, right? One, people who are doing de novo prediction, they just use the mechanics and the physical nature of the protein they have in hand. And one is they learn how certain sequences and certain parts of the protein fold from all the existing structures we have. And 170,000 structures is a pretty big training data set. And over the times, we have also kind of saturated into new kinds of folds we can identify in proteins. So we are almost there in terms of having a complete set. So it's not just a bigger data set, it's also kind of a complete data set that you're not missing out any unknown folds as per se. So I think it's a huge step in terms of this protein folding science, and it might be really useful in terms of drug discovery, because a lot of new diseases and new problems we identify in cells are at the end related to some structural malfunctions or structures misfolding of a protein, right? So if we have a tool in hand that can predict the first draft of the protein structure, that would be really useful. But in a larger picture, I think, like AlphaGo, of course, is big enough to think about, but if you go beyond that, it's still a step, a tiny step where you predict a structure of a protein. And a protein is, I don't know, a millionth, a 10 millionth part of a cell, right? So.
But I think what AlphaFold does is take away, if we could predict even half of, or 3/4 of the protein structures on their own, that takes a big burden away from what all we have to do. But the real key, as it is the same problem in World in a Cell, is being able to predict interactions accurately, because that's how proteins function. They interact with other things and make things happen. So that will be and I would predict that probably, given everything and the power of computing that we have now, probably that'll be able to be done pretty soon too. And now we can just enjoy explaining it all to people, okay? Instead of doing the whole part about getting the structure, figuring out what it looks like, blah, 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 blah. We don't have to do that anymore. So I view, okay, so in my earlier career, I worked in a cancer center, okay? And we worked on cancer. And I remember people saying, what are you going to do if we find a cure for cancer? And I said, hooray, you know, we don't have to worry about that anymore. The same thing here with protein folding. If we get that out of the way and we have an extra tool that allows us to learn these shapes, that's fantastic. So I think that this is a big step forward in what we need to do. So
I mean, it feels like like how, and I think it's coming from how AI is being presented to the world in terms of kind of a unemployment generator or something. But it looks like, oh, okay, what this huge structural biology community going to do now if AlphaFold can predict every protein structure? But I think we were kind of restricted by protein structure prediction. Now we have AlphaFold. It opens up many more doors to move ahead and do the things we were not able to do because we were just spending years and years to predict the structure of the protein.
Great, thank you. That's really helpful for people. The other thing that it made me think, hearing Dennis on the radio, was I remember the first time I met Dennis Hassabis, he was a teenager who was designing a computer game around theme parks. And it's, the transfer of skills is such a, and how you need to bring the right set of skills together seems such a crucial part of any project. And so this is the final thing is the world in a cell. So a crystallographer, I mean, Alex, I think is still listening to us, but was there earlier, you know, a feature film designer, creative technologist in Brian, an architect, Alexandra, you know, molecular biologist. That's an incredible team to pull together. Can you keep it together as a band? I mean, is this how do you do that?
I think we can, because one of the things that happened in the project, and it wasn't just a bunch of people coming together at a certain time every day to do something. We've really bonded as a group, and there's a core of us who, through thick or thin, have made a commitment to stay together and work together somehow, okay, through whatever we have to work. So this is this whole project for people you see here and a few others is really the embodiment of art science, and we see that and we're devoted to it really in our guts now, okay? And that's one of the things that happened by working together, producing this product, and then realizing that there's so much more to go and how much more we can learn if we can just figure out how to do this. And so I think that I'm sure we'll continue to work together and build this. And I think we really have completely concurred about the importance of art science in the doing of this kind of work. So.
Great. All I can say is thank you so much, all of you, for sharing it with us at Beyond. And I know we were all going to be in South by Southwest last year showing Art in a Cell, but perhaps by the time they can accommodate us in real life, we'll be able to show it fully in the round. Thank you so much.
Thank you.
Thank you.